Okay, so we're going to go over cell metabolism. By cell metabolism, we mean how a cell gets energy, are the basics of it. It is actually a very complicated process, and the science of biochemistry is the process by which most of this information has been figured out. So we do have to start with some fundamentals of chemistry, including thermodynamics. And thermodynamics is a branch of chemistry that's concerned with energy. And if you think of a pot of boiling water, or any action really, it requires energy. Usually, the most common form of energy we think of is heat. So heat travels through the flame into your pot of water. It's conducted through a metal pot. There are a bunch of different ways that heat travels then, or energy is transferred. Living things, just like this pot of water, are also constrained by these laws of thermodynamics. And to the left here we have a perpetual motion machine, which we will talk about later. Energy can be broken up into two main components: kinetic energy, which is energy in motion, and potential energy, which is energy that is stored. Now, energy is stored in the food that we eat, or in other macromolecules that we covered in previous chapters. Or also, you can think of potential energy being stored in this roller coaster example. So a roller coaster at the top of the tracks has lots of potential energy. It can be sitting still, not doing anything, but because it's high and the force of gravity will pull on it, excuse me, it has a high potential energy. Once it starts going down the track, then that potential energy is being converted into kinetic energy, and we have energy in motion. Likewise, a person or a rat in a hamster wheel is taking the potential energy stored in the food that it ate and converting it into mechanical energy. It is moving. You could also, and then that's being transferred into, could be transferred into electrical energy if you have a generator. But yeah, those are the basics then: potential and kinetic energy. And energy can take a lot of forms. We already talked about heat. We talked about mechanical energy, which is movement. There's also sound. Electricity is a form of energy. Radioactivity and light are also forms of energy. And energy is often measured as heat. It's just the simplest way to measure it. And we measure it in units called calories or joules, but calorie is one we're most familiar with because that's what we talk about when we talk about our foods. How many calories, how much energy does it have? And 1 calorie is the amount of energy required to increase the temperature of 1 gram of water 1 degree Celsius. Okay, so that's what a calorie is. Now, the source of energy of living things on the planet comes from, of course, the sun. The sun is constantly radiating heat and light towards the Earth's surface, and then that is what sustains life. It is captured by, of course, our plants or photosynthetic organisms, as they, through the process of photosynthesis, they convert light energy into chemical energy. And it's stored in living organisms in our 4 macromolecules, our organic compounds: carbohydrates, lipids, proteins, and nucleic acids. And then we also have ATP, which is how we use it. And that is answer to the last question. And how is it used in living organisms? Through the conversion of ATP to ADP. And we'll talk more about that later. So, chemical reactions have the ability to store or release energy, and this is in the bonds that they create among the atoms within them. We talked about oxidation, reduction, reactions before, or redox, and through a series of redox reactions, our bodies and living cells are able to capture or release energy. Okay? If you remember, an oxidation is when an atom or a molecule loses an electron, and then a reduction is when an atom or molecule gains an electron. So this diagram here shows we have two molecules, A and B. They react with each other, and A donates an electron to B. Because A loses an electron, it is being oxidized. Because B gains an electron, it is being reduced. And the oxidized form of a molecule is going to have more energy. So B, at the end of this reaction, has more energy than A because it has an extra electron that allows it, that bond has energy within it, has more potential energy. Alright, so the first law of thermodynamics refers to energy. And energy cannot be created or destroyed, it can only be changed from one form to another. So remember, we talked about the different forms: light, heat, electricity, mechanical, all those so you can change heat into electricity. But you cannot take an energy and destroy it, or take energy out of nothing. Okay, that's the first law of thermodynamics. So that means that the total amount of energy in the universe is fixed. There's only a certain amount, and it remains constant no matter how many times energy is transformed from one form to another. It's still, the overall energy in the universe remains the same. Now when you do convert energy from one form to another, some of it is always lost. So nothing is 100% efficient. And it's usually lost as heat. So I have a couple pictures here of what are called perpetual motion machines. These are machines designed to show, to go on forever. Basically, defying this first law of thermodynamics by being 100% efficient. Well, these are not exactly 100% efficient. And we have a YouTube video, we will show this in class, of some of these perpetual motion machines and how they seem to defy this first law. The second law of thermodynamics concerns a principle called entropy. And entropy is the disorder in the universe. So, the second law of thermodynamics is that the random order of matter is continually increasing. So things are constantly going from an organized state to a more random state. And that the measurement of that disorganization is called entropy. So in other words, entropy is constantly increasing. This, again, figure shows, if you relate this to your room or your house or your kitchen or whatever, it doesn't spontaneously clean itself. But without very little effort, your room can go from clean to messy. That's just the nature of things. Things don't. And in order to organize it back into a clean space, it requires input of energy. It requires you to actually do something. So the way we measure energy is through the the symbol for it is capital G, which is free energy. And this is the amount of energy available for work. So again, because we said not every conversion of energy is 100% efficient, some of it will be lost. And what is lost is called the entropy. Okay, so our free energy equals the enthalpy, which is another E word. This is the amount of energy within the bonds of a molecule. So that's not including entropy. But we have to subtract our entropy, the amount that's going to be wasted when it's converted, and that is also dependent on temperature. So the amount of free energy equals the enthalpy, so the total amount of potential energy within the bonds, minus the entropy times the temperature. Okay, and we see this when we convert. Here we're converting mechanical. Have you ever tried to start a fire with some sticks? You're converting mechanical energy into heat. Okay, and what you're doing is releasing chemical bonds within your body, causing movement, and then that also causes friction, and then what's lost is heat. Okay, and with enough of that, you can start a fire. We also see this when we are running or when we are exercising. You will start to overproduce heat because when you are converting molecules into energy, a lot of it is lost as heat. Okay, so a change in free energy, this sign, the triangle, is the Greek letter delta, and delta G means change in free energy. Okay, so in a reaction, if the change in free energy is negative, that means the reaction is giving off energy. So that's a spontaneous reaction. That would be like your room just becoming messy. That's just going to happen spontaneously. If the change in free energy is positive, that means it requires energy. Okay, that would be like cleaning your room. It requires energy, and in the end, you're going to have less energy than when you started. So, and that is called an endergonic reaction. When you have negative free energy, ones that give off energy, those are exergonic. We use exergonic reactions to release energy in cells, and then that energy is used for whatever function of the cell. It could be movement, or it could be photosynthesis, whatever. When we want to store energy, we have to put energy into a reaction, and those are usually endergonic reactions. Okay, but in an exergonic reaction, there is a certain amount of energy required to get it started, and that's called the activation energy. Okay, so the rate of the reaction then is dependent on the activation energy, and that's what this graph illustrates. You have products which have less energy than our reactants, so it's going to give off energy, and that's the difference between this line and that line. But there still is a certain amount of energy that you have to overcome before the reaction can complete itself. So to increase the reaction, we can either increase energy, so put more energy into the reactants, usually as heat, or we can decrease the activation energy somehow. And we can do that with the help of a catalyst. And a catalyst is some molecule that affects the chemical bonds in a reaction so that the activation energy is smaller. Okay? But it does not change the products or the reactants, and it does not change itself. Okay? So in this graph, again, you have an exergonic reaction where the products are lower in energy than our reactants. And this blue line is an activation energy without a catalyst. When you have a catalyst, it reduces that activation energy, so the rate of the reaction goes faster. Okay, again, we talked about energy stored in chemical bonds and releasing those to capture energy. We do that through the molecule adenosine triphosphate, or ATP. And all cellular actions require this molecule, or one like it. It is composed of ribose, a 5-carbon sugar. If you remember from our DNA and RNA, they had deoxyribose and ribose, same sugar, adenine, and phosphate molecules. If you have 3 phosphate molecules, it's ATP. And the bonds between those phosphate molecules are unstable, and that's what will release energy when one breaks off. So that's illustrated here. Here's our adenine, here's our ribose, here's our 3-phosphate molecules, and the bonds between them are high energy bonds, meaning they are unstable and they will release energy quite quickly. If there's only 2 of them, then it's called ADP, meaning there's only 2 phosphates, and you also have AMP, or monophosphate, adenine monophosphate. So when an ATP is converted to ADP, it will release energy. Okay, and that's what's used by the cell to fuel it. ATP, however, itself is not, you don't store tons of ATP in your cells because it's so unstable. So when you have too much ATP or you have excess ATP, then you will use that ATP to make macromolecules such as fats, lipids, triglycerides, or carbohydrates. Enzymes are catalysts that are found in living things, and they're usually made of protein. There are some also made of RNA. And what will happen is the enzyme will bind to a substrate. It will lower the activation energy and change the shape of it, or cause stress on the bonds somehow. That will lower the activation energy. And that will cause the reaction rate to go faster. Sorry, my son just came in the door. Alright, again, just like catalyst, it remains unchanged throughout the reaction, and it can be used again and again. So enzymes have certain sites within them, so arrangements of molecules that will bind to the substrate. And when it does that, it's called the enzyme substrate complex. And like I said before, it will apply stress to the bond somehow, change the shape, and this will lower the activation energy. This is an illustration. I'll let you kind of go through this, and we'll actually go through this in class, just illustrating the binding of the enzyme and the substrate. Now, the enzyme usually only works within a range of conditions, and this is important for things like in the human body because different parts of the body have different conditions. So there is usually an optimum in temperature or pH or other things that may affect the rate of the reaction for the enzyme. And we see that here. The optimum for pH for pepsin is around 2. Well, pepsin is found in the stomach, and the pH of stomach acid is about 2. So it's going to work best when it's in the stomach. Trypsin is found in the duodenum, or the smaller intestine, and so the pH there is much higher, closer to 7, and trypsin works best in that pH, so that's where you will find it. Alright, so there are different types of things that will also bind to an enzyme. One is called an inhibitor, and that will increase the activation activation energy or decrease its ability to catalyze a reaction. Competitive inhibitor binds on the same site as the substrate, so if the inhibitor is there, then the substrate cannot bind there. Or a non-competitive inhibitor will bind to another site, and it will cause a change in the active site so that it no longer can bind to the substrate. Okay, an enzyme that has an active and an inactive form is called an allosteric enzyme. And it usually has a molecule that will bind to its allosteric site, and it may inhibit the enzyme, which would be an allosteric inhibitor, or it can also have an activator, which would be an allosteric activator. So the binding of this molecule will increase its ability to function as an enzyme. So finally, we're now getting to the concept then of metabolism after we've gone through the background in chemistry. So metabolism is all of the chemical reactions taking place within an organism. Okay, and you have different types of reactions. You have an anabolic reaction, which is when energy is used to build molecules, so you can build triglycerides or carbohydrates. Catabolic reaction is when things are broken down and energy is released. And a biochemical pathway, can be for the building of a protein, the building of muscle, or it can be for the making of energy or the storing of energy. All of these different pathways have a sequence of enzymes that are used to facilitate reactions, and one product of one of these reactions will be used for the next step. Okay, to regulate the amount of these reactions, or regulate metabolism, you have what's called feedback inhibition. And that's really just where the end product also will bind to an enzyme and will decrease the ability of that enzyme. And in that way, you will not have an overproduction. It will not continue to go forever. So the end product, say this is a protein needed to digest food. It will be activated when food is present. It will go through a biochemical product, and then the end product will bind to the enzyme and decrease its ability to function in the future. So as long as the end product is being used to digest food, so as long as food is there, then it will continue to be produced. But once food is gone, then you will have a buildup of this product, and instead of being used to digest food, it will bind to the enzyme and decrease the production of it. So, respiration then is the most efficient way that we are able to get ATP out of organic molecules. It does require oxygen for most organisms, but there are some other organisms that do not require oxygen and use other molecules instead. Autotrophs are organisms that are able to synthesize organic molecules through the process of photosynthesis. So plants, algae, there's also certain types of bacteria that can also photosynthesize. Heterotrophs are organisms that either eat plants or eat other animals, that get their energy from organic compounds rather than from photosynthesis. So again, cellular respiration and the metabolism of cells depends on a series of redox reactions where certain chemicals are used to convert ADP into ATP. Okay, and what they do is they make a proton gradient. A proton is a positively charged subatomic particle we talked about before. And we'll go more into this in greater detail. Okay, so if you remember, an oxidation reaction happens when a compound loses an electron, and a reduction reaction is when a compound gains an electron. Dehydrogenation is a specific type of oxidation or reduction where a lost electron is also accompanied by a proton or a hydrogen atom. If you remember when we talked about basics chemistry, a hydrogen atom is made of a proton and an electron. So one of these redox reactions is by a molecule called NAD, and NAD plus is an ion that in a reduction reaction gains a proton and an electron, and 2 electrons, and it becomes NADH. So NAD plus is a positive ion, so it gains 2 electrons. If it just gained 2 electrons, it would be a negative ion, but because it also gains a proton, it's a stable compound when it is NADH. When NADH is oxidized, it will donate a proton and 2 electrons to become NAD+. So it is used basically as a electron and proton carrier. Okay, we'll talk more about NAD later. So the sequence of redox reactions in cellular metabolism oxidizes organic compounds. So it takes energy away from them. It takes electrons away from them. And it reduces our electron receptors NAD plus and FAD plus. Okay, FAD plus is very similar to NAD plus. And they become NADH and FADH too. So now they have these hydrogen ions, they have these hydrogen molecules on them. And we have a picture here. This is kind of big and confusing, but basically what to notice here is we have NADH here. And what happens is NADH and also FADH too will let go of their proton and they will send it to the other side of this mitochondria. And through a process of reactions, you will get lots of hydrogen atoms up here. And because of diffusion, they will want to go through back down to the other side through this membrane. And when it does that, it causes this enzyme to convert ADP and phosphate molecules into ATP. Again, we're going to go into this in a lot more detail, but those are the basics of why oxidation and reduction are important to respiration. Now, there are different types of respiration. If oxygen is the final electron acceptor, so going back here, when these protons go through back down the membrane, they convert ADP to ATP, and then also oxygen accepts them and is converted, so that's what happens up here, is converted into water. So if oxygen is the final electron acceptor, it's called aerobic respiration. If they use a different type of molecule, then it's called anaerobic respiration. And if they don't use that system at all, it's called fermentation. And the electron acceptor is an organic molecule instead of oxygen. Alright, so the change in free energy is negative 686 kilocalories per mole of glucose. So you don't have to really know what that is, but you do have to know. I mean, you don't have to memorize that, but you do note that this is an exergonic reaction, meaning it is a spontaneous reaction that gives off energy. And if it's constrained within a cell, then it will be, the change in free energy can be even higher, so you can get even more out of it. But it's a series of very many small steps, not just one big reaction. So we write it out. So this is the process of respiration. You write it out, and you have, this is a sugar molecule, and you have oxygen, and it combines to carbon dioxide, which is waste, and water, and then you get a bunch of ATP out of that. But it's not that simple. There are a bunch of little reactions that have to happen in order for that to happen. Okay, and again, ATP is going to be our energy molecule for inorganic, so using energy, or catabolic reactions. The change in free energy in one ATP molecule is 7.3 kilocalories per mole. So a lot less than aerobic respiration. And there are two mechanisms for making ATP. You have what's called substrate level phosphorylation, and this occurs in a process called glycolysis. Okay, and this transfers phosphate groups directly to our ADP. Oxidative phosphorylation, this is our aerobic respiration, and this is the enzyme ATP synthase, uses energy from a proton gradient. So that's what we explained in that figure before. Again, this is, we're going to go in a lot more detail, but just know you have substrate level phosphorylation or oxidative phosphorylation. So the first step in aerobic respiration is we need to make some molecules for these different cycles. And so what we're going to do is a process called glycolysis. If you remember, lyse means to cut, and glyco refers to glucose or sugar molecules. So we're going to cut a sugar molecule, cut a glucose. And then you have pyruvate oxidation, the Krebs cycle, and then the final is the electron transport chain and chemiosmosis. We're going to go again through these in detail, starting with glycolysis. There are many steps to glycolysis, but the basics are you convert one glucose molecule into two pyruvate molecules. Glucose is a 6-carbon sugar, and pyruvate is a 3-carbon molecule. So you have two pyruvate molecules. In that process, you reduce two NAD molecules into NADH, and you convert two ADP molecules into ATP. So already we are making energy, and we are making electron carriers, NADH. This occurs in the cytoplasm of the cell. Okay, again, net production of two ATP molecules, and this is substrate level phosphorylation. Phosphorylation means you are creating an ATP molecule from ADP and our two NADHs. Okay, so if you want to get energy just out of glycolysis, you have to have NAD+. So in order for glycolysis to continue, you have to convert NADH back to NAD. And that can be done either through aerobic respiration, which also produces a lot more ATP, or if you don't have oxygen, some molecules or some cells, prokaryotes in particular, which are called anaerobic, will convert NAD+ or convert NADH back to NAD+ in a process called fermentation. And that will allow glycolysis to continue, and they'll still get 2 ATP for every glucose molecule it has. Okay? We as humans do this, and it's called lactic acid fermentation. Okay? There's also ethanol that can be created in alcoholic fermentation, and that's used in plants to create alcoholic beverages. Alright, so in order for pyruvate to be oxidized, if we have oxygen, we will do that through the process of aerobic respiration. And it occurs in the mitochondria. So that we have to take pyruvate then from the cytoplasm and transfer it into the mitochondria. And this occurs because prokaryotes do not have a mitochondria. It occurs at the cell membrane in prokaryotes. Then for each 3-carbon pyruvate molecule, you have 1 carbon dioxide of waste, 1 NADH, and 1 acetyl-CoA. The acetyl-CoA is then used in the next step, the Krebs cycle. CO2 is not used for anything, it's waste, and this NADH can then also be used in the electron transport chain with our other NADHs. Alright, so that's all we're going to do for, there's more steps obviously, but that's all we're going to do for today, and we'll see you in class.